Okay, today we'll hear from Andy Moeller, who I think everybody knows. He's a postdoc in my lab. He's a Miller postdoc. He's been in the MVZ for 1 year, and he's been remarkably productive. So in his first year as a Miller Fellow, he published a paper in Science, among other things, which you should check out if you haven't seen it. And he did his PhD at Yale University with Howard Ochman. And today we'll be speaking about some work that he's done since he's been here, based on samples of gut microbes from all sorts of wild mammals. So with that, I'll turn it over to Andy. And his office is right next to mine, right here, if you want to reach him and talk to him.
Thanks. Yeah, I'm glad I'm going before Ruth Lay. She's really fantastic, so everyone should come next week too. So the field of microbiology started with this person here, Antony van Leeuwenhoek, who was a Danish draper or fabric maker. He made clothes. And he was living in the late 17th century, and like many fabric makers of his day, he had an interest in microscopy, because lots of drapers would use microscopes to examine the quality of different threads and fabrics that they were selling or manufacturing. And at one point he got his hands on a book by Robert Hooke that described his studies using microscopes to look at microstructures and even discover cells in organisms, and was really captivated by this. And so Leeuwenhoek already knew how to make microscopes, but he really became obsessed with improving the quality of microscopes and making better and better and better microscopes. And so this is just an example of what his microscopes look like. They're really small, maybe like this big, and you hold them like this and sort of look with your eye, one eye, through the lens here, and you put your sample right here. And the lens is just a small, tiny glass sphere. And Leeuwenhoek's microscopes could get up to something like 300x magnification, even though it wasn't a compound microscope. So it's a really powerful microscope. And he moved on from fabrics to looking at all sorts of things. And he wasn't a scientist. He came from just like a middle-class, normal family. He wasn't like landed gentry or anything like this. But he was very curious, and he would take his microscopes and look at everything he could. So he looked at things like lice. He was the first person to look at sperm and describe sperm motility. I think he was the first person to discover blood cells. So these are actual drawings of red blood cells. And he sent letters to the Royal Society of all of these observations. He never actually published a paper or he didn't know how to write a paper, I think. He was never trained. And his letters are like 30 pages long, just rambling about the weather in Denmark, and then with like a few drawings. But he became really famous. The Royal Society, Robert Hooke, he was a contemporary of Hooke, even said it's a shame that the entire field of microscopy has now come to rest on one person's or one man's shoulders. And his relationship with the Royal Society sort of shifted when he sent them a letter describing what he called animalcules. Which we know now they're single-celled organisms. So he looked in rainwater and saw these little tiny things swimming around, which we know are protists, and he also looked in dental plaques and saliva and found these rod-shaped single-celled organisms, which we now know are bacteria. And when he sent these letters to the Royal Society, they didn't believe this at all. They're like, oh, this is too bad. This person's an insane, crazy person. We've been listening to him all these years. And so they sent Robert Hooke down with a few other people, or up to Denmark, I guess, or east to Denmark, and actually to see for themselves that these things were in fact real. And now he's considered the father of microbiology and sort of the founder of the field. I admit these drawings are not very convincing, right? These are just like anybody can draw them. And so this led to a century, really, of people using microscopes like this and compound microscopes, just cataloging microorganisms in every environment, just looking at every possible surface or liquid and categorizing all the different microbes that were there. And eventually that process transitioned into a more I guess, scientific or process-based understanding of microorganisms and led to the germ theory of disease and a lot of revolutions in modern medicine. So today we're really in a new era of Leeuwenhoek-esque cataloging of microorganisms, but it's one enabled by DNA sequencing. So new Illumina technologies that allow us to look in environmental samples and sequence all the microbes that are present. And it's a really simple process. You just can extract DNA, amplify any regions of interest, and just sequence them. And you can see all of the microbial diversity in your sample. Maybe the pinnacle of this new era of cataloging is the Human Microbiome Project, which is an initiative started in the last few years to, I guess it was almost a decade ago now, but to catalog all the microorganisms in the human body, in every body site. We found that, we knew this already, but the vast majority of microbes live in the gut, and especially in the large intestine. And so we harbor about 40 trillion bacteria, most of which live in the gut, and representing thousands of species. I put this in quotes because it really means a 97% OTU, or a species is defined as any organism that shares 97% of its 16S gene gets lumped into a species. So it's not really a biological species concept, more just a descriptive one. And I think the reason that the human microbiome is getting so much attention and research support is that differences in community composition of the microbiome, particularly in the large intestine, have been associated with a lot of different phenotypes and diseases. So things like diabetes, asthma, obesity, even some behavioral disorders seem to be associated at least with the microbiome. And in some cases, experiments in germ-free mice have been able to show that the microbiome actually has a causative role in some of these diseases, like obesity and even autism. But one problem, I think, is that we don't really understand how the microbiome got to be the way it is, how it evolved. And that really is a limitation due to a lack of knowledge about the microbiomes of non-model organisms. I think that the MVZ is a really great place to start exploring the microbiomes of non-model organisms to try to understand how microbiomes evolve alongside their hosts. So Michael mentioned this earlier, but last year a few months ago, we published a paper that showed that some of the most prominent lineages in the human gut of bacteria can trace their origins to bacterial symbionts that lived within ape populations, ancestral ape populations. So in other words, some lineages like the Bacteroideaceae and the Bifidobacteriaceae have co-speciated with apes and humans. So these phylogenies, each branch is a bacterial lineage, and they're colored based on the host species from which they were recovered. And you can see that in pretty much all cases, the topology of the bacterial phylogeny matches the host phylogeny here, with maybe one exception in Bifidobacterium, where a lineage seems to have jumped from bonobos into gorillas. And we can date the divergences of these co-speciating gut bacteria and find that so this is just using BEAST and some simple molecular clock assumptions. And we find that the divergence times of the Bifidobacteriaceae and the Bacteroideaceae are roughly in line with the known divergence dates of hominids. So the bacteria diverged, we estimated, between 5 and 6 million years ago. The bacteria from humans and chimps 5 to 6 million years ago, and humans and gorillas a little over 15 million years ago. But we also found that other groups of bacteria aren't co-speciating with hominids. So they're being transferred around among host species pretty frequently. So like the Lachnespiraceae, for example, if you look at this phylogeny, there's pretty much no signal of the host phylogeny in this tree. So that indicates this group of bacteria is constantly being transferred among host species. And it's going to come up later, so I just wanted to note that this lineage, or this group of bacteria is spore forming. And so it can form hard spores that help it survive in harsh conditions. And so that might be one difference. So the Bacteroideaceae and the Bifidobacteriaceae aren't spore forming. So it might be whether or not you're spore forming determines whether or not you can jump around between host lineages potentially. So that is just sort of introduction to get to the point that our hypothesis going forward is that mammal gut microbiomes are really composites of co-speciating and independently diversifying lineages. So some gut bacteria are maintained strictly within host lineages, whereas others seem to be able to move across host lineages. And so for the rest of the talk, I wanted to address two basic questions. One, how are bacteria maintained within host lineages? So what are the processes or the circumstances that promote the maintenance of bacterial lineages within host lineages never being transferred between host lineages? And I'll tell three stories, one from chimps, which is work I've done, and then two other stories, one in baboons and one in bees, which isn't work I've done, but other groups, which really great work, I think that, and I think these three stories together tell a cohesive narrative about how bacteria are maintained within host lineages. And then in the last part of the talk, I want to get at the question, by what routes are bacteria transmitted across host species boundaries? So this isn't only an important question for understanding how microbiomes or commensal and mutualistic bacterial communities evolve, but most human pathogens are derived from cross-species transmission events, and as far as I know, we don't have a very good understanding of how that process actually works and what promotes that sort of host switching. So I want to try to answer that question as well, or at least get at it. And something I'm not going to talk about, but I just wanted to mention, hopefully there'll be a free seminar slot next year, and I can come back and talk about what I'm most interested in my postdoc here, which is to try to understand the functional consequences of microbiome differences among host species. And so we're starting some experiments with germ-free mice where we're actually treating different mice with different bacterial communities to look at function, but I don't have any data from that yet, so I'm not going to talk about it anymore. So first, I want to talk about this question: how are bacteria maintained within host lineages? And one obvious, sort of straightforward mechanism that can enable vertical transmission over evolutionary time, or enable the maintenance of bacterial symbionts within host lineages over evolutionary time, is vertical transmission. And so maybe one of the most straightforward examples of vertical transmission of bacteria is aphid Buchnera system. So aphids have this specialized organ called a bacteriocyte that harbors a very interesting bacterium called Buchnera that helps the aphid acquire essential amino acids from its diet. So the aphid feeds on sap, and so it can't get a lot of the essential nutrients it needs to live, and so it relies on this symbiont Buchnera to produce a lot of the essential amino acids that it needs. And Buchnera is vertically transmitted from the mother to the progeny, so there's no external acquisition of Buchnera. It's actually deposited as into the progeny directly from the mother. So this raises the question, what's going on with mammals? Are mammal gut bacteria vertically transmitted? And the answer to this question is pretty complicated, and I think it's actually still somewhat contentious in the field, the degree to which gut bacteria in particular are transmitted vertically from mothers to infants, and whether that is a dominant mode by which new generations acquire their bacteria. So for the longest time, the paradigm was that the womb is sterile and that infants in the womb are sterile. But there's some evidence now that people have actually looked using these culture-independent approaches and found evidence that some bacteria might actually be present in utero. But then even in the last year, there have been some studies saying that, well, the bacteria they're finding actually don't differ from negative controls. And so this is still an open question of whether or not vertical transmission is the dominant route of transmission in gut bacteria. But what we do know is that throughout the first year of life, the development of the gut microbiome is very chaotic. And so I think it's safe to say that the inheritance of the microbiome is more complicated than just strict vertical transmission. Vertical transmission might be a component, but it's much more complicated than that. So this plot is just showing on the x-axis days after birth and a measure of phylogenetic diversity in the gut microbiome of a single infant over about 900 days after birth. So each point represents a fecal sample taken from this infant. And what you can see is that the diversity levels sort of rise and fall substantially over time. and it takes about a year before you sort of plateau out and acquire your sort of stable, healthy adult microbiome. And so during this period, the infant is being seeded by microbes from external sources in addition to the mother. And so it's clear from this alone that you don't start life with your healthy microbiome and mature microbiome. It actually is a process that takes, acquiring it is a process that takes at least a year, maybe even more. So an alternative to strict vertical transmission is social transmission. So this is another route or another mechanism that can maintain microbial symbionts within host lineages over evolutionary time. So you imagine you have an ancestral population here that splits then into two diverging populations. If bacteria are transmitted only within populations through social interactions, but never or rarely between populations, you can still have co-association and co-divergence of bacterial lineages with host lineages. And so this is analogous to how genes diverge between diverging host populations. So the same thing could be going on with microbes. And so I want to talk briefly about a study that we completed last year as well, where we used Gombe chimpanzees to try to understand how gut bacteria are transmitted across host generations. So the benefit of this system is that it's been studied for decades, and all the chimpanzees, not only do they all have names, and we know the ages of all of them, but we know the genealogies of all the different families of chimpanzees that live in Gombe. And we also know through lots of behavioral observation work the social networks and the social structures and interactions among the chimps. And so with those data sets combined, we can tease apart the influences of strict vertical inheritance from social inheritance. And so we collected 160, or we analyzed about 160 samples collected over 10 years from chimps of known relatedness. And so these samples span 4 families, major families in Gombe: the T family, S family, G family, and F family. These are families of chimpanzees. And then we have some samples from other families as well. But this shows the sampling period went from about 2000 to 2009. We have pretty good coverage of all these families over this time period, although there are some gaps. But so what we found was that over time, within a social group, that the compositions of chimpanzee gut microbiomes change in parallel. So in panel A, each point here is a sample from a different sample from an individual chimpanzee. And the points are colored based on whether or not they were collected in the first half or second half of the sampling period. So the point of this figure is just to show that there was a shift over time in the composition of the community, that all the chimpanzee microbiomes shifted in parallel over the sampling period. So that the distances among chimpanzee microbiomes within seasons were generally less, significantly less than they were between seasons. So microbiomes were more similar in the same season than they were between seasons. And also that we could see this compositional shift evident in infants that were sampled over this time period as well. So this plot is just showing infants sampled in the first half and second half of the study period. And we also found that the differences among microbiomes within a maternal line were not significantly different than the differences between microbiomes between maternal lines. So it looks like the microbiomes of socially interacting unrelated chimpanzees are just as similar as the microbiomes of related chimpanzees in the same social group. So this already suggests, to me at least, that vertical and social transmission are both contributing probably, but social transmission might be contributing just as much as vertical transmission to the composition of the chimpanzee microbiome. Yeah, so that's just... so to really get at whether or not social interactions are promoting exchange of bacteria, we can take advantage of the fact that chimpanzees display these seasonal cycles in social behavior. on the y-axis here, we have the half-weight association index, which is basically a measure of the amount of time any two random chimpanzees are seen interacting or in the same area. And so bigger numbers mean that the chimpanzees are more socially active, they're forming larger groups and hanging out with each other more often, whereas lower numbers indicate that chimpanzees are spending more time alone or in small social groups. And the bars represent either dry season or wet season in each year. And so you can see that in each case, in each year, dry seasons are always, chimpanzees are always less social in dry seasons than they are in wet seasons. So in dry seasons, food is less abundant, and chimpanzees are traveling long distances to find food and not spending as much time together, whereas in wet seasons, they sort of huddle together in the same group. And so in addition to social data, we also have dietary data from this throughout this sampling period. And so we can look at the effects of social interactions and diet together and sort of control for both of those to try to assess whether or not social interactions are promoting exchange of bacteria among chimps. We found that across this sampling period that chimpanzee microbiomes are more compositionally similar during periods of high social activity. So on the x-axis is this measure of sociability, the half-weight association index, and as chimpanzees become more social, the differences on the y-axis between their microbiome is measured by this ecological metric of binary break curves, or Sorensen-Dice metric. We see that as chimpanzees become more social, their microbiomes become more similar. So each point is a pairwise comparison between two chimpanzee microbiomes. So there's a lot of variation here. The R-squared is pretty low. And so it's not explaining all of the variation but among chimpanzee microbiomes, but there is a definite trend here that suggests that chimps are exchanging bacteria when they're interacting socially. We also found that chimpanzee microbiomes are more species-rich during periods of high social activity relative to periods of low social activity. So again, on the x-axis here is the half-weight association index, the average half-weight association index of the chimpanzee population, and this is just a plot, or the y-axis is indicating the number of observed species in individual chimpanzee microbiomes. And we see a kind of a subtle trend that as the chimps are interacting more, their microbiomes become more species-rich, more diverse. And this is we can also control for the effects of dietary variation. So an alternative explanation might be that chimpanzees are also eating more variable diets during periods of high sociality, so that might confound our conclusions that social interactions are driving us. But we can use mixed modeling approach to actually disentangle these two and show that both the diversity of the chimpanzee diet and the frequency of social interaction are seem to be exerting independent effects on microbial diversity, or at least are independently associated with microbial diversity. But what we'd really like to be able to do, which we couldn't do in this study, is to ask whether or not social networks among chimpanzees can predict gut microbiome composition. So we didn't have quite the resolution in our data set to do this, but this study that came out last year from Jenny Tung and Elizabeth Archie at Duke and Notre Dame, respectively, studied baboons and was actually able to get at this question. And so I'll just briefly go over this study as well. So they're studying baboons living in Kenya in two different social groups, which they're calling Mika and Viola. And they found that the different social groups harbor compositionally distinct gut microbial communities, even though they're eating basically the same diets. And they were actually able to show through sampling the feces of the baboons and also a year of behavioral observations that the strength of grooming relationships between baboons actually predicts the compositional overlap between their gut microbiomes. So in A and B, these are social interaction networks or grooming networks between baboons and the Mika and Viola social groups. And so the thickness of the lines indicates the strength of the grooming relationship. So thick lines mean like F28, F29 are grooming each other a lot, whereas F28 and F27 almost never groom one another. And you see this pretty strong or pretty good negative correlation between the distance between microbiomes and the strength of the grooming relationship. So the more frequently baboons interact, the more similar their microbiomes become. In this stage too, they were able to identify bacteria that were socially structured. So these are bacteria that show the strongest signals of being determined by social interactions. And they found that these bacteria tend to be non-spore forming. So like the Bifidobacteriaceae and the Bacteroideaceae that have co-speciated with apes and humans, the bacteria that are socially structured in baboons are non-spore forming and actually include the two groups of bacteria that seem to be co-speciating with humans. And they don't include the Lachnospiraceae, which seem to be jumping around among the species. So to me, this is at least consistent with the idea that social interactions might be the key to maintaining these lineages over evolutionary time. And I think it'll be really interesting to look at all of these other groups that they identified and also look at a broader range of host taxa to see whether or not these are co-speciating with hosts as well. So just briefly, I wanted to mention too that social transmission has also been implicated in the maintenance of bee gut symbionts over time. So social bees harbor a very simple gut microbiome that consists primarily of 8 species. And these 8 species are not found in asocial wasps. Asocial wasps have very variable microbiomes, don't really seem to have a core microbiome, but bees do. And in fact, some of these lineages, these core 8 phylotypes, have co-speciated or co-diversified with Aphis and Bombus, so honeybees and bumblebees, excuse me. And a lot of great work by Nancy Moran and her group, Walvin Kwong, and other people have shown that social transmission is really the main route by which these symbionts are maintained over time. So the bees are born sterile, and they're only colonized after adult emergence. And it takes a few days to establish this characteristic bee microbiome. And the transmission routes are primarily fecal-oral, directly from hive material, and through oral trophallaxis. So interactions among bees are really the key to maintaining these symbionts within bee lineages. So I think these 3 studies and others that are coming out are suggesting this general rule that social behaviors might be essential to the preservation of microbial symbionts within animal lineages in the absence of strict vertical transmission. So the vast majority of symbionts we know aren't strictly vertically transmitted. And so I think social behaviors might really be the key to this. And so it's interesting that to think about how bacteria and other symbionts might actually be influencing behavior, social behavior. So we know social behaviors influence symbionts, but are they actually promoting certain social behaviors for their own interests? So, why do something like mouth kissing in primates, which is, why does this happen? Maybe the bacteria are making us do this, I don't know. So in the last part of the talk, I want to talk about work that is done in collaboration with a lot of people in the museum, trying to get at this question of what processes promote the transmission of microbes between host species. So I think social transmissions are key to maintaining lineages within host species, but what about transmission between host species? And we don't know the answer to this question really, because the landmark surveys of mammal microbiomes, which studies, one of which was performed by Ruth Lay in 2008, these taught us a lot about mammal microbiomes, but they weren't really designed to examine interspecific bacterial transmission. They relied primarily on animals living in zoos and captivity and isolation from one another. And the studies of wild mammals that have been done have typically focused on just individual clades of hosts living in different geographic regions and haven't been able to look at interspecific microbial transmission. So really, to get at this question, what we need to do is to study the microbiomes of entire assemblages of hosts at once, and see, in order to understand the routes by which microbes are transferred. So to get at this question, we collected a big data set of mammal fecal samples from throughout the Americas. So I'm sorry this is very faint here. But yeah, so we have samples from mammals ranging from South America all the way up to Canada. From this list of species we'll go through quickly: wolves, coyotes, bears, cougars, bobcats, caribou, deer, moose, elk, tucos, voles, grasshopper mice, deer mice, kangaroo rats, pocket mice, chipmunks, and squirrels residing throughout the Americas. This is in collaboration with Eileen, Dana, Michael, Taichi, who are all in the museum, and Sam Wasser at University of Washington. So all these people provided studies and helped a lot with thinking about this project. So I'll just present some sort of general results from this work. And one observation that jumps out right away is that mammal gut microbiomes diverge compositionally alongside host lineages. So on the x-axis here is divergence time between host species. Each point represents a pairwise comparison between the microbiomes of two host species. And the y-axis indicates the compositional overlap between gut microbiomes. And so you can see that as evolutionary divergence between the host species increases, the compositional overlap between the microbiomes decays exponentially with time. And another thing that jumps out is that Sympatry slows and or reverses microbiome divergence. So this dark gray line and the dark gray points represent comparisons of host species living in Sympatry in the same community, whereas the light gray points and the light gray line correspond to host species living in allopatry. And so using this exponential decay model, we can actually put a half-life on the compositional overlap between diverging host species. And so when species live in allopatry, it looks like the compositional overlap between their microbiomes is cut in half about every 15 million years of divergence. But when the host species are living in Sympatry, that half-life goes up to about 50 million years. So it really slows down the divergence between their microbiomes. But we'd really like to be able to answer the question of what are the routes by which gut bacteria are transferred between species. And so to do that, we can leverage our data set, the fact that it includes samples from lots of host species that all live in Sympatry together in northeast Washington. So this is a set of species that are all found in the same geographic region. So cougar, bobcat, wolf, coyote, deer, moose, elk, pocket mice, chipmunks, squirrels, and deer mice. And this figure is just showing the predator-prey relationships, or the preferred predator-prey relationships between these different groups of hosts. So the large-bodied carnivores, cougars and wolves, tend to prey on large-bodied artiodactyls, whereas the small-bodied carnivores focus more on rodent prey. And so the expectation then is that if predator-prey relationships are promoting the exchange of gut bacteria, that cougars and wolves should share more with artiodactyls than do bobcats or coyotes, and bobcats and coyotes should share more gut bacteria with rodents than do wolves or cougars. So we can actually test this with our data set, and we find that in every case it looks like predator-prey relationships are promoting the transmission of gut bacteria across those species boundaries. So the way to read these figures, each bar represents a pairwise comparison. So this, for example, is between cougars and elk, bobcats and elk, and so on. And so I'm just going to run through all of these results, but the point is that cougars and wolves are more similar to this artiodactyl prey species than are bobcats or coyotes. So this is compositional distance between microbiomes. And that's true for Cervicelaphus, for moose as well, for deer, And if we look at the rodent prey items, we see the opposite pattern, where bobcats and coyotes share more bacterial lineages with things like Paramiscus than do cougars or wolves. And that's also true for chipmunks, for squirrels, and for pocket mice as well. Although this comparison, although it's in the direction we would expect, it's not significantly different. And so you could think of maybe some alternative explanations for this pattern. One is that it could be driven by body size effects, because predators, small body predators tend to feed on small body prey items, large body predators tend to feed on large body prey items, so maybe that's driving the pattern. So we can test that, whether that's going on, by comparing, I don't have figures to show this yet, but we can look at large body carnivores living in Washington and compare them to large body prey items. So we have samples, for example, from caribou living in the northern part of Canada that aren't sympatric and never preyed on by the wolf populations that we have. And when we do that comparison, wolves and coyotes versus those caribou, we actually don't see the same pattern. And the same thing for rodents living allopatrically from the cougars, wolves, bobcats, and coyotes. We don't see the same trends in those comparisons. So that suggests it's not driven by body size effects. And another possible caveat is that maybe these prey-derived phylotypes are just transient and passing through the gastrointestinal tract of the carnivores and not actually stably colonizing and proliferating within carnivores, which would, I think, make it much less interesting. But I don't think that's going on either, because we can find phylotypes derived from multiple prey species present within individual carnivore fecal samples. So the fecal samples we're using, we'll take an aliquot, maybe a tiny, 0.1 gram, and we'll find prey-derived phylotypes or bacterial species from many species of prey, all within that one fecal sample. So carnivores, as far as I know, people here could correct me if I'm wrong, but it's not going to be very common that a wolf would feed on a moose and then immediately feed on an elk and then a deer all on the same day within the time it would take to pass that one fecal sample. So I think that suggests that these phylotypes are actually stably maintained within host lineages. So yeah, I just wanted to conclude that social relationships unite the microbiomes of host populations into what we're calling pan-microbiomes. And that pan-microbiome, it's really a meta-community of microorganisms that is evolving alongside host lineages. So these meta-populations are evolving alongside host lineages. And that the differences among microbiomes of host populations and species can break down due to ecological interactions among host species, especially those that promote direct contact like predator-prey relationships. I wanted just to pitch or to make the point that the museum here is really well suited to understand and reconstruct the evolution of vertebrate microbiomes. And so there's a lot of projects going on that aren't yet finished, so I haven't had a chance to talk about them. But using this microbiome tissue collection that started in 2015 and that's ongoing and growing, I think it's going to be a really valuable source of data for the field, not just for people working here, but potentially for researchers all over the world. Yeah, so I just wanted to thank Michael and the Nachman Lab and my PhD committee who contributed to this work, and some collaborators as well and funding. So thank you.
Yeah, the similarity indices you showed were statistically obviously very significant, but some of them differed in the third decimal place.
Yeah.
Is that biologically significant?
Yeah.
Or is that what you're going to tell us next year?
Yeah, so the communities consist of thousands of species, or in some cases thousands of species. And so differences even in the third decimal place can represent tens of species that are shared. So it is in some cases it's a very small effect. In some cases it's hundreds of species that seem to be shared between predators and prey. So I think, yeah, the question is whether or not, what effect do those species have on the recipient hosts, on the predators that receive them? And that, we don't know the answer to that. That would be interesting.
The thing that struck me was how close to one all of those indices were.
Yeah.
Which somewhat, I don't know, was that surprising to you, or?
It's pretty typical, I think, in this field to see, even within human populations that could be sampled, everyone in this room, like a break courtesy, the break